Hello guys, so today we are going to see action potential of the neuron. This is going to be our topic first. So let's just see the diagram, and from that we will understand how this neuron and the membrane, the connection, the exchange between them happens. That will be here through a diagram than to then further coming this graph and then this other diagram. So here it is. Now what we need to see here is this is the membrane, the upper part, and this is the neuron we have here. Okay. So in a neuron we have charges, and even on a membrane we have charges. So this is a neuron and this is a cell membrane. Now we have charges on a cell membrane. Okay. And what kind of charges are they? They are potassium, they are sodium, and then we have some organic acids containing some charge as well. And because of these charges present outside the membrane, we call it that the neurons also have some charges. And what is basically they're trying to say is that neurons also have some of having charges, they have some potential. So that's how they say neuron action potential. Now action is something else, but neuron potential, so this is the potential because of the cell membrane having charges, and the neuron says I also have the potential of doing the same. And also the membrane having potential is called the membrane neuron membrane potential. All right. And when there is potential of the membrane here, we can see, then of course there going to be electric potential between them, the neuron and the membrane. They're going to be electric potential, right? And this potential is going to get noted by volt. And in a neuron we always have potassium ions more inside the neuron. Okay, yeah. So outside we have sodium ions, and they are like 10 times more than the ions we inside the neuron to be the sorry the which is going to be the potassium inside the neuron, and sodium ions um, outside the membrane. And that's what I said, that is going to be 10 times more than the ions we see inside, that is the potassium. And if sodium ions are like 10 times outside than the potassium inside, then the potassium inside is like 20 times more than the sodium ions outside. Okay, so this is all about the this is all about the potential of the neuron. Now let's see what is the resting membrane potential. We need to know that too, like the membrane potential. So the membrane potential is going to be when the neuron is more negative inside than the outside. Like the so resting membrane potential is when there's more negative ions inside. And if the ions are like positive inside, then it be a resting membrane going to be the conduction. So it means that the neuron is conducting the stimulation and the conducting the charges, the current, or you can say energy. Now, what can be the thing which is making the neuron negative inside? What are the things? The very first thing is that the sodium potassium pump. Now, in sodium potassium pump, what happens that you can see here the sodium and the potassium. So the sodium ions goes three sodium ions goes outside, and in return two potassium ions comes in. So now you can guess that three ions of sodium are going out and the two potassium ions are coming inside. So what is going to happen now? Now you can yourself guess that if the three are going and inside we have two, so which side is going to be more negative? Of course, outside. We will see the more positive ions, and inside we will see the less negative ions. So if the less positive ions we are seeing inside, it means that it is like more negative as compared to the outer part, which is like having more positive ions. So the inside view is going to be negative. So clearly by that we can see this is one of the reasons the sodium potassium pump is responsible for making the neuron inside more negative when we are talking about the resting membrane potential. So this is what is happening here. That's why it's being negative inside. Although if you see there's no negative charge inside, but because there's less positive ions inside, we get a negative portion as compared to the outside one. Another reason to make it more negative is going to be the organic acids we have inside the neuron. They're also going to cause it to be the negative. Then we will have the third one is going to be the potassium leakage. It happens because you can see here that the membrane is leaking the potassium, because the membrane is like more permeable to potassium ions than to sodium when it comes to the when it comes to resting membrane thing. So again, what is going to happen here, if the potassium ions are getting leaked out, then what is going to happen? Again, we will see the less positive ion inside. All right, and when we have less positive ions inside, what do we call it? We call it negative. So again, these three are going to be the three reasons why neuron is being negative inside. The very first one, let's recall, it's going to be the sodium potassium pump. The second one is going to be the organic acids or the protein, and the third one again the potassium D. And when neuron is that much negative, we call it, what do we call it? Let's just say it, resting membrane potential. So here we have the first potential, the membrane potential. Okay, now let's come back to the neuron. So what do you want to do with the neuron now? You want to see its action. So for the action, what we are going to do first, we are going to provide it the energy, and that energy we are calling it as stimulus. So here now what the stimulus is going to cause a disturbance in the ion system. And because of this distance, we can clearly see that here, the sodium ions here are going to get inside, and the potassium ions are going to go outside. And here the depolarization will happen. It's going to cause the depolarization, and here the sodium ions are going to going to activate um, the gate, and the gate is going to get open, and now what is see is that the sodium ions are flowing inside. But before getting into this, we need to know what is depolarization is something we gave the stimulus. Now that stimulus is going to create the disturbance, and due to the disturbance, what we will see is here the charges are going to get differ. Now the positive charge is going to get in and the negative outside, because you can see the sodium ions are getting inside, right? So it's being now less negative, and when it's being less negative, then what is it going to be? It is going to be the positive one, and outside is going they are coming inside. So what outside is like less positive, and less positive is negative. Inside it's more positive, so it is like the positive. So that is going to happen, and then the charges like this. Oh, yeah, positive here, and it's going to be the negative, and again, yeah. So this is happening. This is going to happen. So this phase is going to be the depolarization phase. Let me write it. This is so now after the depolarization, what will we see is that they're going to be the same thing. The nerve impulse is going to, yeah, the nerve impulse is going to get along and it's going to run now. So we can see it coming here. The same thing is going to happen. It will also get negative outside and positive inside, because the charges are getting continued and the impulse we can is getting. So this is what is going to happen. But now there is that this portion is also going to get depolarized, right? And the other portion is the stimulus is going to run the impulse. We are going to get the in the long run, the impulse wherever it's going, it's getting depolarized, doing the neuron depolarized. But the area which is getting behind, like this area here now, let me take another color. Now this area here, because now this portion is getting depolarized, right? Now this one is also getting depolarized. Now this area which is leaving behind is not going to get called depolarized, but it is going to get repolarized. So this area is going to be, so now when this area is getting repolarized, what is going to happen, that the charges are going to the same in the original, and they are going to be like that. The inside is going to be the negative charge again, and the outside is going to be the positive charge again, as it was before before the stimulus. So this is what is called repolarization, and then we have this is what the depolarization. So depolarization is the first, but when the depolarization will start to get impulse forward, the left ones, the before the ones which are at the back are being called repol, because they are getting repolarized. They are coming to their back polarized situation. And here the current is going forward, but behind it is getting neutral as it's starting to get forward. So now we can see what is an action potential. The action potential is the action potential here. It's actually the depolarization of the neuron or the membrane, which simply means that inside there are going to be more positive ions. Now let's see the same in the diagram, the repolarization, how it's happening. So now you can see here the sodium ions are going outside. They're taking themselves outside. That's how it is getting negative again inside and positive outside. Now let's see this graph. It's time by membrane potential. So here we have the resting potential, which we already studied now. It's in rest, and now it's getting the threshold. What is threshold? Okay. So it is the minimum energy required to start any nerve impulse. That's how the potential is. So the threshold potential is here. Now it's getting that required energy. The stimulation is happening to the resting potential, the resting membrane. Now it is getting that threshold, which is that energy required for the nerve impulse. So the nerve impulse is going to happen, and it's going into that membrane potential thing. It has reached its most potential. Now again it's coming back to that resting potential. Now you can see it's coming back. Now when it's coming back to the resting potential, it's getting more like down than the resting potential, which is minus 75, negative 75. It is getting more negative. So the when it hits the membrane potential, and when it's going back to the resting phase, it gets more negative as before. You can also see the potassium relative permeability. So this permeability is more when it's at the resting potential, when it's getting into the resting when it's getting from the resting potential to the threshold. You can see the membrane is being more permeable to potassium ions. as they have mentioned it here also. It's more permeable to potassium than to the sodium ions when at rest. And as soon as it goes to that potential membrane, it reaches the high potential. Now we can see here, we can see more sodium permeability than the potassium one. Now let's see anything again. So what the book says, let's see it. So it says that membrane repolarization happens when the sodium inactivation basically closes the gate. So when it was closing the gate, so here we have the stimulus and here we have this peak. Here we have this peak potential. When this peak, when the potential is coming down of the peak, the sodium inactivation happened, the gates, and thus this causes sodium ions to stop flowing that way, and it start to inflow, like inside it start to get in. And whereas we can see that the potassium ions now their gate is getting open, due to that the potassium is flowing outside. So what we will see here again, the in repolarization, the inner neuron part is going to be negative and the membrane is going to be the positive again, because the gate has been closed and now we can see the sodium is flowing into, which is making it or opening the gates for the potassium and through which it's going outside. So that's how it's again getting negative inside and the positive outside. Now we have the membrane hyperpolarization. So what was the membrane hyperpolarization? We actually saw it before. Now this is our this is our we gave the stimulus. This is our membrane at rest. Okay. Now this hyperpolarization is when it's coming backwards to the repolarization, and after the repolarization, when it's going again at the resting membrane, it's going little bit more down and it's get more negative. It's getting down towards more negative value, and thus it's making inside of the cell more negative than the resting state. All right. Now, now due to this hyperpolarization thing, what happens is due to this shift in the cell membrane potential and that negative value we are getting more, makes it more difficult for an action potential to be generated. Now the next action potential is going to be harder to be generated, and this is how it's going to inhibit the neural activity and it's going to prevent the continuous firing of action potential. And this is how we will see cells excitability and their function. Now let's look at the sensory receptors. So in sensory receptors, we have these type of receptors, which are five. So let's see the first one, the free nerve ending. So at the free nerve ending, we will find these more subtypes of receptors, which is A delta, C, and A delta. Yeah, these two types. Okay. So in A delta is going to be fast and it's going to be myelinated fibers. Okay. If the fiber endings are going to be myelinated, then it means that the conduction velocity is going to be higher in that, and the C type is going to be low and it's unmyelated, so the conduction velocity is not there. It will be there but less. So and also we need more time. Sorry, just to yeah. So the unmyelated one, we will need more time to depolarize it. That's why it's going to be slower. And now they are giving us a mnemonics here, which means that A delta, like they just giving example, delta plane is fast, but a taxi is slow. So that's how you can maybe remember it. Now, where we are going to find it is in all tissues except the cartilage, eyelids, and skin. And what senses you will feel by that is pain and temperature. So you can also remember it like that. That's AC. by AC. So when we feel cold due to AC, what we do we feel often is pain and temperature. So that's how you can remember. That's how you can remember it. And what where we going to feel the pain? We're going to feel the pain in almost all the tissues except where we not going in the cartilage, eye lenses, and other than that, you will feel everywhere and mostly in the tissues. Now we have meninal carpuscles. They are going to be large. Let's just assume that um, they are some kind of flowers. It's a kind of mnemonics you can remember by that. So my Y and E got kind of mixed. You can get it. Or maybe I should just write it again. So yeah, you can assume them as like lovers. So what happens? The lovers be doing a lot of promises, right? So they speak like they're gonna do a lot and this and that. So they are going to be large and myelinated. Again, the conduction velocity is going to be higher and quickly. They do promises quickly. So yeah, adopt quickly. Than the skin, they will have a glabrous skin and so that they'll have a fine touch, dynamic. They'll feel a fine touch. They'll feel dynamic and they're going to be low. So usually when the lovers are sitting, they be talking in low voice, right? So that's how you can just assume it that low frequency vibration going to be sensed and skin indentation is going to be sensed, like depression or hollowness of the skin. Now we will see the Pechian one. So here you can take it as same as the lovers one only. You can take it as example like that. So it's going to be easier. So again they're going to do what? They're going to do large, big conversations. They are same as myelinated fibers and they're going to do promises quickly. So like that. And now they have started to fight and so we will see high frequency vibration and pressure. So apart from the example, these are the senses we are going to feel. This receptor is going to make is that high frequency vibration is going to be felt by this, and the pressure is going to be felt by this Pacinian. All right, and where it's going to be? It's going to be in the deep skin layers. All right, and it's going to be in the ligaments and the joints. Now we have Michael Dix. So, this type of receptors are again going to be large, myelinated, adopt, but they're going to adopt slowly. Okay, so this is something you need to remember. We don't have any example or kind of mnemonics here. So, that's how it's happening. Now, where it's going to be? They're going to be in the fingertips. They're going to be in the fingertips and they're going to be in the superficial layer of skin. Okay. Now, what they're going to sense, Michael Dix, what they're going to sense, they're going to sense and deep touch. Here we had the fine one, and here we have the deep static touch. Like what are those sharp touches, like sharp edges, like a needle or pen kind of. Now, but something to not forget is that in Michael Dix, they're going to adopt slowly. Now we have Raffeni corpuscles. Now here what you will see, we will see it that they are also like large, right? Myelinated fibers, but there's something different is here. Now they're not getting solely adopt, they are actually intertwined among the collagen, like they're interconnected in collagen inside the collagen fiber bundles. Okay, they are all connected and they are going to be adopt slowly like this, Michael, this okay. But only the new things being added here is they're going to be intertwined among the collagen fiber bundle. That's what. And the same we are going to have them in the fingertips, at the fingertips and the joints again. Joints we had it here also. Okay, now what they're going to sense, they're going to sense the stretch and the joint angle change. So that's what. Now something to see here, that these what they're going to do here in the A delta, they're going to sense the cold, okay, and these C type, which is flow type, they're going to sense the hot temperature. So basically, it's the temperature they're going to sense, and they're going to feel the pain. Sense actually. So this is all about these sensory receptors now, and let's just end this lecture here. So that's it.